Blood typing in the lab involves mixing blood samples with various antiserums. In the lab, we have anti-A, anti-B, and anti-RH antiserums. Anti-A antiserum contains antibodies against A-type antigens. Anti-B antiserum contains antibodies against B-type antigens. And anti-D or anti-RH antiserum contains antibodies against RH antigens. If the blood sample clumps after a few minutes after it has been mixed with the antiserum, it means that particular antigen is present on the erythrocyte surface. If the blood sample does not clump after a few minutes after it has been mixed with that particular antiserum, it means that that particular antigen is absent from the erythrocyte surface. In our lab, the blood typing cards we have look like this, where there are 3 sets of partially overlapping circles. In the circle on the right of the overlapping circle set, a small drop of sample blood is placed. On the left side of the overlapping circle set, a single drop of each corresponding antiserum is placed. From these 3 antiserums, we can use the results in order to determine which of the 8 blood types, along with the RH factor, that a particular person has. Now, we'll go through a few specific examples to see exactly how this works. This blood typing card looks a little different than ours, but the basic principle is still the same. What we see is that one small drop of blood from the same person has been placed in each of the 4 circles. In the circle to the left, the blood sample was mixed with anti-A antiserum. In the second circle from the left, the sample drop of blood was mixed with anti-B antiserum. In the circle second from the right, we see that the sample drop of blood was mixed with anti-D or anti-RH antiserum. And in the circle on the far right, we see that the sample drop of blood has been mixed with a control antiserum. Now, in the control antiserum, we should see no clumping occur under any circumstances. So if the blood were to clump in the control antiserum, this means that the blood needs to be retested with fresh antiserums, as incorrect results would be obtained from this particular test. As we see, no clumping occurred in the control, so that means that the results from this test can be interpreted as accurate. We do see clumping in the anti-A and the anti-D antiserums, but we do not see clumping in the anti-B antiserum. So this means that this particular sample is A positive blood type because we had clumping in the anti-A, which means that the A antigen is present, and we also had clumping in the anti-D or the anti-RH antiserum, which means that the RH factor is also present. In this sample, we see that no clumping has occurred in the control sample, so that the results from this test can be interpreted as accurate. We see clumping has occurred in the anti-B and the anti-D or the anti-RH antiserums, but we see that no clumping has occurred in the anti-A antiserum. So this means that the sample is B positive blood type. In the last example here, we see that no clumping has occurred in the control sample, so that the results can also be interpreted as accurate from this particular test as well. We do see that clumping has occurred in the anti-D antiserum, but it has not occurred in the anti-A or the anti-B antiserums. So what this means is that the sample is the O positive blood type because the sample did not clump in either the anti-A or the anti-B antiserums, which means that the A and the B antigens are absent, and therefore the only remaining antigen is the O antigen.